Hi guys, welcome to the IBuddies YouTube channel. And here we have a DNA model that is 3 base pairs long. You can see that the orange balls are phosphates and they're covalently bonded, as you can see here. All the white bonds are covalent. So they're covalently bonded to the ribose sugars. They're also bonded from the 5 carbon to 3 and the 3 carbon. So this strand here goes from 5 carbon to 3, and then the opposite strand goes in the opposite direction. So it goes from 5 prime to 3 prime. The ribose sugars are also covalently bonded to the nitrogenous bases. And here we have adenine and guanine, which are purines. And as we know, the purines are too big to be bonded together. So a purine has to bond with a pyrimidine. And in this case, it's adenine bonding with thymine. And when adenine bonds with thymine, there's only 2 hydrogen bonds, as you can see here. Whereas when guanine bonds with cytosine, there's 3 hydrogen bonds.
So hi guys, so this is a more magnified version of what Aref recently explained. So first we start off with the replication bubble, and this DNA helicase unzips this DNA strand and that creates a replication bubble, and this is another one right here. So DNA gyrase is used for stability, and it prevents the DNA from supercoiling. And DNA helicase, as I previously mentioned, unzips the DNA. And we're going to start off with the leading strand. So it goes from a 5' to a 3' direction towards the replication fork. And this right here is the parent strand, the yellow, and the daughter strand is the blue. So the green is the polymerase 3, and that's what puts the DNA nucleotides, the complementary ones. And this blue thing right here, that's the DNA polymerase 1, and that proofreads what polymerase 3 does. And this is important because it prevents mutagens and yeah.
Okay. And then you have the lagging strand. So this isn't smooth and continuous just like the leading strand because it goes away from the replication fork. Because the parent strand is going from 5' to 3'. And because of this, you need to use many more enzymes and proteins to have the replication process occur. So first, as you can see, these in both strands you have the single binding proteins, and what these are for is just to stabilize the replication process. And then it starts with a primase enzyme, and what this does is that it adds RNA primers, which are about 10 nucleotides long, to the strand. And this is basically just to start the the replication process so it can occur. And then after it adds primers, it continuously adds primers in fragments called Okazaki fragments. And after it adds them in fragments, DNA poly 1 comes in and removes the RNA nucleotides, because this is a DNA replication. So it removes them, which is the blue thing, DNA poly 1, and then poly 3 comes in and it replaces them with DNA nucleotides. So the nitrogenous bases that Aref explained, G and C, A and T. And then after you're done that, you need to join these fragments together, or else it's not going to be a continuous strand. And that's what this duct is for. And this duct is DNA ligase. And as I just mentioned, it just joins the Okazaki fragments together so it can create a continuous strand. So another key component that you guys need to know about DNA replication is that it's semi-conservative. So after Martha Chase and Alfred Hershey's experiment with the nitrogen mediums and the isotope of N15, they have proved that DNA is actually semi-conservative. So what this basically means is that if you have if you just have a parent strand, after it replicates, you're just gonna have a strand of new, so the green being the strand of old, and then the pink being a strand of the new. And then you have the old, and then you have the new. So that's why you have some characteristics from your dad and some characteristics from your mom. And that's why every human is unique, because the DNA replication is semi-conservative.